Chapter 3, Plant Kingdom. In the previous chapter, we looked at the broad classification of the living organism under the system proposed by Whittaker's son 1969, wherever he suggested the 5 kingdom classification: Monera, Protista, Fungi, Animalia, Plantia. In the chapter, we'll deal in the detail with further classification within kingdom Plantia, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and member of the Monera and Protista having cell wall have now been excluded from the Plantia through earlier classified placed them into the same kingdom. So the cyanobacteria that are also referred to as the blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophyta, pteridophyta, gymnosperm and angiosperm under Plantia. Let us also look at classification within angiosperm to understand some of the concerns that influenced the classification system. The earliest system of classification used only gross superficial morphological characters such as habit, color, number and shape of leaf. They were based mainly on vegetative character or the androsium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to the vegetative and sexual characteristics. That is not acceptable since we know that often the vegetative characters are more easily affected by environment, as against the nature classification system developed which were based on natural affinities among the organisms and considered not only the external features but also internal features like ultrastructures, anatomy, embryology and phytochemistry. Such as classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestors. We now use in information from many other sources to the help resolve difficulties in classification. This become more important when there is no supporting fossils evidence. Numerical taxonomy, which is now easily carried out using computer, is based on observational characters. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance. At the same time, hundreds of the character can be considered. Cytotaxonomy, that is based on cytology information like chromosome number, structure, behavior and chemotaxonomy that uses the chemical constitutions of the plant to resolve confusions, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll-bearing, simple, thalloid, autotrophic and largely aquatic, both freshwater and marine organisms. They occur in a variety of the habitat, moist stone, soil and wood. Some of them also occur in association with fungi like lichen and animals. Sloth bear. The form and size of algae is highly variable, ranging from colonial form like volvox and the filamentous form like Eurothrix and Spirogyra. A few of the marine forms such as kelp from massive plant bodies. The algae reproduce by vegetative, asexual and sexual method. Vegetative reproduction is by fragmentations. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores. The most common being the zoospore. They are flagellated and motile. When germinated, gives rise to a new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eurothrix, or non-flagellated and non-motiles, but similar in size as in Spirogyra, such as reproduction in called isogamous. Fusion of a two gametes dissimilar in size as in species of Eudorina is termed as angio and isogamous. Fusion between one large non-motile female gamete and a smaller motile male gamete is termed as oogamous, like volvox and fucus. Algae are useful to man in variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthesis, they increase the level of dissolved oxygen in their immediate environment. They are the permanent importance of primary production of energy-rich compound which form the basis of the food cycle of all aquatic animals. Many species of Porphyra, Laminaria, Sargassums are among the 70% of marine algae used as a food. Certain marine brown and red produce large amount of hydrocolloids, water holding substance like algin, brown algin and carrageen, red algin, which is used commercially. Agar, one of the commercial product obtained from Geridium and Giardia, is used to grow microbes and in preparation of ice cream and jellies. Chlorella, a unicellular algae rich in protein, is used as a food supplement even by space travels. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. Chlorophyceae. The member of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass-green due to the dominance of pigments chlorophyll A and B. The pigments are localized is definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in the different species. Most of the members have one or more storage bodies called pyrenoid located in the chloroplast. Pyrenoid contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made up of an inner layer of a cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentations or by formation of a different type of spore. Asexual reproduction is by flagellated zoospore production in zoosporangia. The sexual reproduction show considerable variety in the type of formation of sex cells, and it is maybe isogamous, anisogamous, or oogamous. Some commonly found green algae as a Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Chyra. Phaeophyceae. The member of Phaeophyceae are brown algae are found primarily in marine habitat. These show great variation in size and form. Their range from simple branched filamentous form ectocarpus to profusely branched form as represented by kelp, which may reach a height of so meters. They possess chlorophyll A and C, carotenoid and xanthophylls. They vary in color from olive green to various shaped or brown depending upon the amount of the xanthophyll pigment and fucoxanthin present in them. Food is stored as a complex carbohydrate which may be in the form of laminaria and mannitol. The vegetative cell have a cellulosic cell wall usually covered on the outside by a gelatinous coating of algin. The protoplast contain in addition to plastid a certainly located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place the fragmentation. Asexual reproduction in the most brown algae is by biflagellated zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, oogamous. Union of gametes may take place in water or within the oogonium. Oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common form as Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. Rhodophyceae. The member of Rhodophyceae are commonly called red algae because of the predominance of the red pigment are phycoerythrins in their body. Majority of the red algae are mainly with greater concentration found in the warmer area. They occur in both well light regions close to the surface of the water and also a great depth in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organizations. The food is stored as a Floridian starch, which is very similar to the amylopectin and glycogen in structures. The red algae usually reproduce vegetatively by fragmentations. They reproduce asexually by non-motile spore and sexually by non-motile gamete. Sexual reproduction is oogamous and accompanied by complex post-fertilization development. The common member of Polycyphenia, Gracilaria, and Giladium. 3.2 Bryophyta. Bryophyta include the various mosses and liverwort that are found commonly growing in moist shaded area in the hills. Bryophyta are also called amphibians of the plant kingdom because these plants can live in the soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in the plant succession on bare rock soil. The plant body of Bryophyta is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the Bryophyta is haploid. It is produces gamete, hence mainly plant body of the Bryophyta is haploid. It produces gamete, hence it is called a gametophyte. The sex organ is Bryophyta are multicellular. The male sex organ is called antheridium. They produce biflagellated anthrozoids. The female sex organ are called archegonia, is flask-shaped and produced a single egg. The anthrozoid is released into water where they come into contact with archegonia. An anthrozoid fuses with the egg to produce the zygote. Zygote do not undergo reductional division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not a free living but attached to the photosynthetic gametophyte and derived a nourishment from it. Some cells of the sporophyte undergo reductional division to produce haploid spore. This spore germinate to the produce gametophyte. Bryophyta in general are of little economic importance, but some mosses provided food for herbivorous mammals, birds, and other animals. Species of Sphagnum a mass provide peat that have long been used as a fuel and as a packing material for transshipment of living material because of their capacity to hold water. Mosses, along with the lichens, are the first organism to colonize rock to hence are great ecological importance. The decomposed rocks making the substrate suitable for the growth of a higher plant. Since mosses form dense mats on the soil, they reduce the impact of falling rains and prevent soil's erosions. The bryophytes are divided into liverwort and mosses. Liverwort. The liverworts grow usually in moist, shady habitats such as bank of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of liverwort is thalloid, marcantia. The thallus is dorsoventral and closely appraised to the substrate. The leafy members have teeny leaf-like appendages to row on the stem-like structures. Asexual reproduction in liverwort takes place by fragmentation of thalli or by the formation of specialized structures called gamma. A singular form gamma. Gamma are green multicellular asexual buds which develop in small receptacle called gamma cups located on the thalli. Develop in a small receptacles. The gamma becomes detached from the parent body and germinate to the form new individually. During sexual reproduction, male and female sex organ are produced either on the same or different thalli. The sporophyte is differentiated into foot, seta, and capsule. After meiosis, spores are produced within the capsule. The spore germinate to form free-living gametophyte. Mosses. The predominant stage of the life cycle of mosses, the gametophyte, which consists of two stages. The first stage is the proteinema stage, which is developed directly from a spore. It is a creeping green, branched, and frequently filamentous stage. The second stage is the leafy stage, which is developed from the secondary proteinema as a lateral bud. They consist of a developed from the secondary proteinema as a lateral bud. They consist of a upright cylinder axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bear the sex organ. Vegetative reproduction in mosses is by fragmentations and budding in the secondary proteinema. In sexual reproduction, the sex organs Antheridia and Archigonia are produced at the apex of the leafy shoots. After fertilization, the zygote develop into a sporophyte consisting of a foot, seta, and capsule. The sporophyte's masses is more elaborate than that is liverwort. The capsule contains spores and spores are formed after meiosis, and meiosis have a elaborate mechanism of spore dispersion. And common example of mosses as a funaria, polytrichum, and sphagnum. 3.3 Pteridophytes. The pteridophyte included horsetails and fern. Pteridophytes are usually for medicinal purpose and as a soil binders. They also frequently grow on ornamentals. Evolutionarily, they are the first terrestrial plant to possess vascular tissue, xylem, and phloem. You shall study more about the tissue in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil condition. You may recall that in bryophytes, the dominant phase in life cycle is the gametophyte's plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stem, and leaves. This organ possesses well-differentiated vascular tissues. The leaves in pteridophyte are small microphyllous as in Selaginella or large macrophyllous as in ferns. The sporophytes bear sporangia that are subtended by a leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structure called strobili or cones. The sporangia produce spore by meiosis in spore mother cell. The spore germinate to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic thalloid gametophyte called prothallus. This gametophyte require cool, damp, and shady place to grow because of the specific restriction requirement and the need of the water for fertilization. The spread of a living pteridophyte is limited and restricted to a narrow geographical regions. The pteridophyte bear male and female sex organ called Antheridia and Archigonia respectively. Water is required for the transfer of antherozoite to the male sex gamete released from the Antheridia. The mouth of Archigonia. Fuses of the male gamete with the egg present in the Archigonium result in the formation of a zygote. Zygote thereafter produce a multicellular well-differentiated sporophyte, which is the dominant phase in the pteridophyte. In majority of the pteridophyte, all the spores are the similar kinds, such as plants is called homospores. Genera like Selaginella solvinia, which is produced two kind of spore, macro and micro. Spores are known as heterospores. The megaspores and microspore germinates and give rise to female and male gametophyte respectively. The female gametophyte in the plants are retained on the parent sporophyte for variable period. The development of the zygote into young embryo takes place within the female gametophytes. This event is a precursor to the seed habit, considered as an important step in evolution. The rhodophyte are further classified into 4 classes: Psilopsida, Psilotum, Lycopsida, Silegilena, and Lycopodia, and Spinopsida, Equisetum, and Pteropsida, Dryopteris, Teres, and Adiantum. 3.4 Gymnosperm. जब gymnosperm are plant in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seed that develop post fertilizations are not covered, are naked. Gymnosperms included medial-sized trees or tall trees and shrub. One of the gymnosperm, the giant redwood tree Sequoia, in one of the tallest tree species. The root are generally taproot. Root in some genera have fungal association in the form of mycorrhiza pinus, which in some other cycas small spliced root called coralloid root of the association with the N2 fixation cyanobacteria. The stem are unbranched cycas or branched pinus or candrus. The leaves may be simple or compound. In cycas, the pinnately leaf present for a few years. The leaf in gymnosperm as well as adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. This thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspore and megaspore. The two kinds of spore are produced within sporangia that are borne on sporophyll, which are arranged spirally along on axis to form lax compact strobili or cones. The strobili bearing microsporophyll and microsporophylls are called microsporangiate or male strobili. The microspore develop into a male gametophyte generation with a highly reduced, and it is conflict to only limited number of cells. This reduced gametophyte is called a pollen grain. The development of a pollen grain takes place within the microsporangia. The cones bearing megasporophyll with a ovule of megasporangia are called a megasporangiate or female strobili. The male or female cones or strobili may be born on the same trees like pinus. However, in cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelope, and composite structure is called ovule. The ovules are born on megasporophyll, which may be clustered to form the female cones. The megaspore mother cells divide into meiotically to form 4 megaspore. One of the megaspore enclosed within the megasporangium develop into a multicellular female gametophyte that bear 2 or more archegonium or female sex organ. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophyte and pteridophyte, in gymnosperm, the male and female gametophytes do not have independent free-living existence. They remain within the sporangia, retains on the sporophyte. The pollen grain is released from the microsporangium. They are carried in the air current and come in the contact with the opening of the ovules born on megasporophylls. The pollen tube carried the male gametes grow towards the archegonia in the ovules and discharge their content near the mouth of the archegonia. Following fertilization, zygote develop into embryo and ovule into seed. These seeds are not covered. 3.5 Angiosperm. Unlike gymnosperm, where the ovules are naked, in the angiosperm are flowering plant, the pollen grains and ovules are developed in the specialized structure called a flower. In angiosperm, the seed are enclosed in fruit. The angiosperm are exceptionally large group of plant occurring in wide range of habitat. They range in size from the smallest wolfia to tall tree of a eucalyptus at over so meters. They provide us with fodder, fuel, medicines, and several other commercial important products. They are divided into 2 classes, the dicotyledons and monocotyledon. The dicotyledon are characterized by seed having 2 cotyledon reticulate venations in leaves, tetramerous or pentamerous flower, having 4 or 5 member in which floral hole. The monocotyledon on the other hand are characteristic by a single cotyledon seed, parallel venation in leaf, and trimerous flower, having 3 member in each which flower holes. The male sex organ in a flower is the stamen. Each stamen considered as a cylinder filament with an anther at the tip. Within the anther, the pollen mother cell divide by meiosis to produce microspore, which mature into pollen grain. The female sex organ in a flower is the pistil. Pistil consists of a swollen ovary. It is at its base a long cylinder style and stigma. Inside ovary, ovules are present. Generally, each ovule has a megaspore mother cells that undergo meiosis to form 4 haploid megaspore. 3 of them degenerate and 1 divide to form the embryo sacs. Such as embryo sac has a 3-celled egg apparatus or 1 egg cells and 2 synergids and 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuses to produce diploid secondary nucleus. Pollen grain after dispersed from the anther is carried by wind and various other agents to the stigma of the pistil. This is termed as a pollination. The pollen grains germinate in the stigma and resulting pollen tubes grow through the tissue of stigma and style and reach the ovules. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of male gametes fuses with the egg cell of synergids to form with the egg cell syn gamete to form the zygote. The other male gametes fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus. Because of the occurrence of the two fusions, syngamy and triple fusion, these events in termed as a double fertilization. As event unique to angiosperm, the zygote develop into the embryo with 1 or 2 cotyledon, and the PEN develop into endosperm with providing nourishment to the developing embryo. The synergids and antipodal degenerates after fertilization. During these events, this ovule develop into seed and ovaries develop into fruit. The life cycle of an angiosperm is shown in this figure. Plant life cycle and alternation of generation. In plant, both haploid and diploid cells can be divided by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gamete by mitosis. This plant body represent a gametophyte. Following fertilization, the zygote also divided by mitosis to produce a diploid sporophytic plant body. Haploid spora are produced by this plant body by meiosis. These in turn are divided by mitosis to form haploid plant body once again. Thus, during the life cycle of any asexual reproduction plant, this is an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant group as well as individually represented them differ in the following pattern. Sporophytic generation is represented only by the one called zygote. There are no free living sporophytes. Meiosis in the zygote result in the formation of a haploid spore. The haploid spore divide mitotically and form gametophyte. The dominant photosynthetic phase in such plant in free living gametophyte. This kind of cycles is termed as haplontic. Many algae such as Valvax, Spirogyra and some species of Scleromidominus represented this pattern. Extreme, the type wherever the diploid sporophyte is dominant. Photosynthetic independent phase of the plant. The gametophyte phase is represented by the single and few celled haploid gametophyte. This kind of life cycle is termed as a diploid. Like example, algae ficus represented this pattern. In addition, all seed bearing plants, gymnosperm and angiosperm, follow this pattern with some variety wherever the gametophyte phase is few to multi-celled. Bryophyta and pteridophyta interestingly exhibit an intermediate condition, haplodiploidy. Both phases are multicellular, however they differ in their dominant phase. A dominant independent photosynthetic thyroid or erect phase is represented by a haploid gametophyte, and it is alternates that the short-lived multicellular sporophyte totally or partially depend on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular sporophytic autotrophic independent but short-lived haploid gametophyte. Such as pattern is known as haplodiploidy life cycle. All pteridophytes exhibit this pattern. Interestingly, which most algae genera are haplontic. Some of them such Ectocarpus, Polycypania, kelp are haplodiploidy. Fucus are algae is diploidy.